why don't we move on to our next speaker? And I'm happy to introduce Alessandro Satta. He is a postdoc currently, or a research fellow, sorry, at University of Padua, Italy, but he has come from around Spain and Italy and yeah, all around the world. And I think you're in Italy right now, if I recall. So I'm very happy to have you join us and to hear about the research that you're doing with respect to metabolic engineering and systems biology.
Hello, everybody. Yeah, actually done my work in Australia, University of Queensland in Brisbane. And first of all, Danny, that was a great presentation. I really like it. Yeah, I will start sharing the screen of my presentation. Okay. Can you see it?
Not yet.
Not yet. So, okay, let's see.
It looks like it's starting now.
Okay. All right.
There it is. Great.
Okay. So yeah, hello everybody. Thanks, of course, to Lisa for inviting me to talk in the symposium. Really appreciate it. As Lisa said, I'm Alessandro Satta. I'm currently a research fellow at University of Padua in Italy. In this brief presentation, I will talk about how I use the BioCyc database and how it helped me in my work about terpenoid synthesis in cyanobacteria, work that I carried on during my PhD at University of Queensland in Australia. I will so very quickly introduce the topic, and then I will show you also very quickly how I use the BioCyc database for some bioinformatic works and how it helped me in my research. So, as you may know, so terpenoids are a large family of metabolites essential in all living organisms. They play a significant role in primary and secondary metabolism, in cell metabolism, and industrially they are relevant because they can potentially be used in many industrial sectors, such as pharmaceutical, in agriculture, in the flavor industry, and so on. In plant chloroplasts and in most bacteria, and cyanobacteria as well, they are synthesized through the methyl erythritol phosphate pathway, MEP pathway, which is highlighted in yellow. The enzymatic steps of this pathway, or the MEP pathway, finally produce two C5 molecules, which are isopentenyl pyrophosphate and is isomer, dimethyl pyrophosphate, the MAP. And then the carbon skeleton of terpenoid is built via the prenyl phosphate metabolism, which is highlighted in blue. Here, specific enzyme called prenyl transferases made consecutive condensation of the product of the MEP pathway, so the C5 molecule, APP and MAP, to generate the carbon architecture of terpenoid. So the molecule created in the prenyl phosphate metabolism will then be converted to the equivalent terpenoid classes by specific terpene synthesis. For instance, C10 backbone, so geranyl pyrophosphate, GPP, for instance, will become monoterpenoid. C15 farnesyl pyrophosphate will become triterpenoid, while carbon 20 molecules such as geranyl pyrophosphate are the substrate, the starting substrate for the formation of diterpenoid and tetraterpenoid, such as carotenoid and other photosynthetic pigment. My research focused on prenyl phosphate metabolism, so the part square in blue. In particular, I characterized the prenyl transferases of the model organism, of the model cyanobacterial organism, Synecocystis 6803 and Synecoccus elongatus 7942. So, while in plant chloroplasts and cytosol and also in other bacteria, different classes of prenyl transferases, so GPPS, geranyl pyrophosphate synthase, farnesyl pyrophosphate synthase, and geranyl pyrophosphate synthase play a role in the biosynthesis of different classes of terpenoid. In cyanobacteria, only one prenyl transferase class is present, and it is encoded by the gene CRT. So we basically solve the crystal structure of both enzyme, SYCRT for Synecocystis and CECRT for Synechococcus elongatus 7942. We saw that structurally both enzymes are very similar to each other, as also very similar to those of coming from plants, and we characterize their activity in vitro. Synecocystis CRT in vitro produces a broad range of different chain length prenyl phosphate, so GPP, FPP, and GGPP, while Synechococcus elongatus CRT produces only C20 GGPP. So we can confirm that this one was like geranyl pyrophosphate synthase. So while I was studying these enzymes, I wanted to have a look at their position and genomic context, because exploration of neighboring genes can give you insight, into functionally associated genes and help you to hypothesize whether these can interact to give a specific biological function. To do that, I used the BioCyc database. So I noted immediately that the open-reading frame encoding the CRT of Synechococcus elongatus 7942, so the blue pentagon, is located at the 5' end of a 2-gene operon that include the gene SYMPCC 79420775, that one on the left of CRT. I will now call it PAP2 gene, because otherwise it's too long and complicated. This gene putatively encoded for divergent type 2 phosphatidic acid phosphatase, PAP2, in fact. The protein was annotated as a membrane protein of 155 amino acid in length, and this specific function has not been previously characterized. Enzymatically, this type of phosphatase can cleave nucleoside diphosphate and may participate in the synthesis of phospholipids and tocopherols. Using the amino acid sequence alignment option in BioCyc that I actually squared in red, I noted that this protein is present in several Cyanobacteria species and in other bacteria that actually I didn't show in this table. After having detected this gene, I wanted to understand if this genomic organization, so the two-gene operon, was also conserved in other cyanobacteria. To do this comparison, I used the option of BioCyc for searching orthologous in multiple databases, again squared in red, which is located in the operation option. So what I've done here, I basically manually added other cyanobacteria species for the comparison. For some space reason here, I only showed you a few Cyanobacteria species. As you can see for the table on the right, some Cyanobacteria species possess this operon composed by 2 genes, while other only have 1, so they don't have this operon. For instance, Synecocystis that only have the gene CRT and not the possible putative phosphatase following it. In fact, this genomic organization seems to be more present in the order of Synechoccales, specifically in the family of Synechocassae, which is the family of Synechococcus. However, it is absent in other family. For instance, in the Merismos pediaceae, we see this, which is the family of Synecocystis. So then, using the multi-genome browser option, I also retrieved the entire genomic context of cyanobacterial species compared in the table, and which, in my opinion, makes the comparison graphically, more eye-catching and to be honest, a very useful tool for creating figures to be inserted in possible manuscript. As you can see from the picture, the transcriptional unit of CRT and PAT2 is squared in blue, while it is absent in other species. In this case, for instance, Synecocystis at the end. Given the particular position of this gene encoding a phosphatase, a putative phosphatase, following a CRT gene encoding for an enzyme synthesizing diphosphate molecules, I wanted to understand if the product of those genes, so CRT and PAP2, could cooperate. So we hypothesized that PAP2 might act on geranyl pyrophosphate produced by the gene, the enzyme CRT and the gene CRT to generate the C20 diterpenoid derivative alcohol geranyl. First of all, I tried to purify this enzyme, this putative phosphatase, but I failed in doing it, possibly because it was a membrane protein, and generally those are very difficult to purify. So I tried, therefore, establish an in vivo assay in E. coli to check, to partially characterize it. Because E. coli does not naturally produce geranyl pyrophosphate, C20, because does not possess geranyl pyrophosphate synthase, and therefore it cannot produce also geranyl C20, which is the alcohol that came from geranyl pyrophosphate. The entire operon of CRT and PAP2 and the individual genes, so CRT and PAP2, were cloned separately into a vector and transformed in E. coli. The empty vector serves, as a reference, and basically what we detected here is that the geranyl was detected only in cell expressing the operon CRT and the PAP2 gene, confirming that the PAP2 is an active phosphatase capable of hydrolyzing geranyl pyrophosphate into the corresponding alcohol geranyl. We didn't do the experiment because actually we didn't have time to do that in Synechococcus, but it may be possible that this kind of mechanism can also happen in Synechococcus elongatus. First of all, there are no study on production of geranyl in Synechococcus elongatus, and it may be that this kind of intermediate can be used in Synechococcus for the production of tocopherol and other fatty acids. So to sum up, I think my research is a good example how to use the BioCyc database, even if you have little experience or as a bioinformatic, for create data using this kind of database, and literally with a couple of clicks, a couple of steps, I was able to detect an operon of the gene I was studying, which is the CRT of Synechococcus elongatus, and I was able to identify a gene that has not been previously studied in Cyanobacteria. And finally, I used this information to partially characterize its activity in vivo as geranyl diphosphate phosphatase. So this is my very brief presentation. So thanks for your attention. If you have any question, I'm here for you.
Thank you so much, Alessandro.
You're welcome.
That was an amazing advertisement for BioCyc, but also great, very interesting work that you've done with characterizing a new gene. That's awesome. I think if people have questions, you could go ahead and put them in the chat. But why don't we move on to